Welcome to Ask the Expert. We've got Dr. Jason Bini from Yale joining us today. The title of the talk is Quantitative PET Imaging in Humans of Pancreatic Beta Cell Mass and Function in Vivo. And just a short bio on Jason. He earned his master's degree in biomedical engineering at Stevens Institute of Technology and then subsequently completed his PhD in biomedical engineering at the City College in New York. City University of New York in 2015, and his dissertation focused on using novel clinical imaging systems to measure atherosclerotic plaque metabolic activity, and he's currently using positron emission tomography or PET imaging to investigate these mechanisms of disease and receptor enzyme pharmacology in metabolic diseases such as diabetes and obesity. Current projects include using PET to image dopamine receptors in the pancreas, both in type 1 and type 2 diabetes, whole body distribution of the cortisol activating enzyme 11B-HSD1 in individuals with obesity and NAFLD, and method development for whole body kinetic modeling of novel PET radioligands in clinical and preclinical studies. Thank you so much, Jason, for joining us, and happy to see you with the familiar East Rock in the background there from New Haven.
Yes, thank you. Thanks for the introduction, and excited to be here. Dr. Wesley's done a lot of connecting a lot of researchers throughout the type 1 diabetes field, so it's an honor to be here as one of the next Ask the Experts.
Thank you.
So we'll just do a brief kind of introduction to set the stage here. And a lot of this research and a lot of the talk in the type 1 diabetes field is trying to measure this beta cell mass versus function, and particularly in the early stage of the disease where there's either rapid or slow progression to diabetes. So just to start with kind of a cartoon, and then I'll show some data after this, but even in the normal islet, you have this kind of increased and decreased demand. So when we do these kind of functional studies to measure proinsulin or C-peptide or other functional measures, these can kind of change with the day-to-day status of the beta cells and the patient themselves, where that overall islet mass kind of stays the same. And then, of course, in diabetes, that will be reduced. And that change and reduction is variable among the first year or so in type 1 progression. And so that first vulnerable stage is really what we want to try to understand with some of these tools. And like I said, some of these functional tests don't reflect accurately sort of the reduction. We get an idea, but a lot of times that functional test is too late and it's already full on kind of symptomatic type 1 diabetes. And eventually, if these kind of imaging devices are useful, we may be able to actually measure pharmacological treatments or transplantation of the beta cell mass in the future. But right now, we really want to focus on understanding that first year within diagnosis. And one of the studies I like to use to kind of show concrete example of the variability is you have these patients that all have the same function, and this is measured when they were alive, and then these are actually post-mortem beta cell areas or ex vivo mass that they've measured. And so you can see that even with the same function, they have increasing mass. So this shows again that maybe some of these subjects can have some kind of treatment that can stimulate this mass that has similar function, but maybe there's more beta cells that are not functioning can be increased at some point. And a recent study from Dr. Sims and colleagues really tried to show that some of these predictors now, looking at proinsulin and C-peptide and the proinsulin to C-peptide ratio, you can actually sort of understand a little bit of the stressed versus non-stressed beta cells and kind of that vulnerable stage where proinsulin maybe reflects these stressed cells that can no longer properly function and secrete insulin. So we see those as the high proinsulin more reflective of a more rapid progression to type 1 diabetes, where a low pre-treatment ratio has kind of a slower progression. But can we really use imaging then to combine with some of these measures and try to understand some of these functional measures compared to our imaging? And interestingly, really to emphasize some of the really complexities of the disease is you see the top arrow pointing here to placebo and a low proinsulin C-peptide. So even without treatment to prolong the healthy beta cell population, they have a higher type 1 diabetes free proportion than those who were on the drug but had a high proinsulin C peptide ratio. So there's a lot of stratification here that we really need to dig in and understand beyond just some of these peripheral markers measured in blood, how this is actually happening. So that leads us to how can we use PET imaging as a tool to understand disease progression? So I'll show a little bit of background of kind of where we are at this point with some of these tools, and then kind of lead towards where we would like to go next. And so just to get everyone on the same page when we're talking about positron emission tomography or PET, we take a subject and we lay them in this ring of a camera. And I'm showing the liver here just because it's easy to visualize. And we inject them with a radioactive drug. And that drug is designed to go to a specific target. So in this case, we'll just say we're going to use glucose. So we have radioactive glucose going to the liver, and you're going to get a positron that's emitted from this radioactive drug. It annihilates with an electron, and we get this. The math and physics actually work out beautifully so that 2 photons are emitted in opposite directions, 180 degrees. And for every one of those, we can accurately detect within the scanner where that's coming from. And with some fancy reconstruction images, we can then create this kind of whole body image of radioactive drugs. So in this case, you could just imagine glucose is a high metabolism thing. So you have the brain, the heart, and then this is an example of some tumors in the armpit there. But in our case, we're looking at not just glucose metabolism. We want to label other drugs as radioactive and target specific receptors. And so to do that in the pancreas, one of the ideas is that beta cells are really only 1 to 3% of the pancreas. So we're not going to be able to image those at a single islet resolution, because the resolution of some of these cameras is 2, 3, 4, or 5 millimeters, depending on the camera. And so we're not going to get that islet resolution. However, if these are targeted towards just the beta cells, and the target is expressed only in the beta cells, we know all that signal from the pancreas is just in the beta cell. So it's really about designing those drugs to hit those receptors specifically, not necessarily looking for image resolution in size, but actually target resolution. So we want to hit that target specifically. And we really want to target both active and dormant cells. So we want a receptor system that's going to be still there on the dormant beta cells, even though the cell might not be fully functional. And of course, it has to have high affinity for the target. You can imagine, say, dopamine receptors. There's 5 different dopamine receptors. We want a specific dopamine receptor out of those 5. We want high affinity to one of them, not all 5 of them. So that's just an example of what I mean by high affinity target. And so some of the work that has started with this, of some of my colleagues in the past, they realized that beta cells and some of the neurologic tissues have common cellular receptors and transporters. And a lot of this has to do with, you have the same transporters to either secrete insulin or neurotransmitters. So a lot of this machinery is recycled in the pancreas to release insulin. And by taking advantage of this, we can look at some tracers that have been used in the past. So you can imagine serotonin precursor. Colleagues from Europe have shown that is a good candidate for showing healthy and type 1 reduction. You can see the kind of disappearance of that banana-like structure in the pancreas in type 1 diabetes. Our own group here has shown the vesicular monoamine transporter. And this is actually on the vesicles that secrete insulin. So it's more of a measure of the vesicles within the beta cells. But we show a similar reduction in type 1 diabetes that other groups have shown with other tracers as well. And then more recently, colleagues again from Europe have shown GLP-1 to be useful to measure glucose variability between high and low glucose variability. So there's a collection of tracers here that all sort of share the same story, where we're seeing a large reduction in type 1 diabetes, but still some residual binding, which we think could be non-functional beta cell mass. So there's preliminary evidence that these are useful for receptors and transporters. The one thing about there's no brain penetrant GLP-1 PET ligand yet. So in the case of or VMAT2, it originally started as a Parkinson's tracer. There is some interest in looking at the pancreas-brain connection with some of these tracers to see how that progresses in type 1 and type 2 diabetes. And I'm going to explain a little bit later, sort of rethinking the role of VMAT2 beyond just beta cell mass, and maybe looking at a slightly different story in the beta cell. But first, I'll start with a recent story of sort of how we've looked at a new tracer for beta cell mass. So again, because they share machinery with the neurons, the same ones that are in the islets, we went back and looked at some of the neuroradioligands we had already here at the Pet Center approved for human use, and we crossed that with a gene expression database for the islet. And so we came up with 2 different dopamine receptor ligands. One is this cortisol activating enzyme inhibitor, a serotonin transporter, which you saw from the serotonin precursor. You'd expect that to be there. And then a synaptic vesicle protein, which again is on that islet vesicle that secretes insulin. And so we have 6 different tracers in these groups, and we scanned a male and female from each of these tracers, and it turned out that the one that we saw the most uptake and highest affinity for the pancreas was this dopamine 3 receptor ligand, and it has a 50-fold affinity for dopamine 3 over 2. So that's what I had mentioned before about really trying to find a high affinity target. And then when we went back and looked again at our genetic database, we saw that there was almost no expression in the exocrine pancreas, and again in the other endocrine cells of the islet, mostly limited to the beta cells themselves. So we think this is a good marker for beta cells. And you can see that where these other 2 tracers in images B and C have a higher affinity for D2. And so you can see that it's not really a specific target in the pancreas, so there's a lot less uptake there. But the participation of dopamine in glucose and insulin dynamics has been known for nearly 50 years. And if you look back in some of the papers in the 70s of dopamine infusion, you get illicit responses from glucose, insulin, and glucagon in the pancreas. And then there's actually a lot of studies showing antipsychotic drugs, which are dopamine antagonists, actually causing type 2 diabetes. So there's some kind of trigger there in the brain and the pancreas that ends up causing this beta cell failure, either directly in the pancreas or in the brain. And then actually a agonist dopamine drug can actually prevent type 2 diabetes. So there's something here working centrally and peripherally in the dopamine system. And the ongoing sort of idea here is that L-DOPA is actually taken up into the beta cell where dopamine is synthesized. There is the machinery inside these beta cells to synthesize dopamine. It's taken up by the VMAT2 transporter, which we had mentioned earlier, and it's co-packaged with insulin. So this dopamine and insulin is actually co-secreted out of the beta cell, and that in turn acts on the beta cell itself with dopamine receptors to regulate insulin secretion. And we've shown a little bit of data here. This is just showing in a healthy control on the left. You can see dopamine 3 on the top row and dopamine 2 on the bottom row. And we do see dopamine 3 and 2 co-localized to insulin only, where we don't see any other co-localization in other endocrine cells. And then in this one example of the type 1 diabetic we actually had no insulin and no dopamine in those beta cell or in those islets. So we see both of those are disappearing. So that's a good sign that if there's no beta cell there, we're not getting that signal. So it should be a good measure of beta cell mass. We then did a cross-sectional study of healthy controls in type 1 diabetics. You can see the healthy controls on the top have a very nice high uptake with the green arrows. And then in the bottom row, you can see that reduced when you get into type 1 diabetes. And we quantified that, and it's about a 40-50% reduction. So we expect some of that residual beta cell mass, and these are in long-standing type 1 diabetics. So these are at least 10 years after diagnosis. So there's still some residual binding there that we think is residual mass. And it's variable, which we measured looking at mass and function here. So if you look at some of the C-peptide measurements that we did, you can kind of break this down into 3 groups here. And it goes back to what we want to measure before. You have this mass, which you're measuring, but how much of that is active and dormant? So pairing this with some of these functional measures really gives you a lot of information. So you may have this subject down in the bottom, which has no mass and no function, someone in the middle range with mass and a little bit of function, and then someone in the top that has a very high beta cell mass, but we measured no C-peptide, so no function of those beta cells. And so that can really guide us in really understanding the biological variability of the disease as it progresses. If you can take these kind of functional measures and really measure the mass, you can follow this progression in the first couple of years and kind of tease out how much of that beta cell mass is functional versus non-functional or active and dormant. And importantly, we looked at this again. We looked at duration of diabetes was not correlated with our PET tracer. However, for type 1 diabetes, the age at diagnosis, which tends to be more severe when younger and more acute beta cell death, was very significant with our PET tracer. So we really think we're measuring these more severe phenotypes with our PET tracer, because the folks who had the lowest beta cell mass were diagnosed at the youngest age. And that tracks well with the general hypothesis in type 1 diabetes. And then we also looked at, similar to other recent studies, pro-insulin and C-peptide. And this really starts to highlight some of the interesting things we can look at, because our PET tracer correlated with both of these. But if you look at the C-peptide on the right, you have a bunch of people stacked at 0. So there's a non-functional population. They also have measurable pro-insulin. So that shows you can spread them out a little bit, and you can actually see that there's variable mass and variable pro-insulin secretion. So there's a population here which we can tease apart, which others have done in larger studies. Colleagues at the University of Florida have showed the same thing, where you see this spread of pro-insulin that really mirrors kind of what we think of as a population of non-functional beta cells. And then in the C-peptide measurements, you really have that, just everyone's at 0 for C-peptide. So that's kind of, you're getting a little bit more information with that pro-insulin measurement versus the C-peptide, and our PET measure actually reflects that as well. And so just to go back to the VMAT2 story, more recent data really showed that VMAT2 is co-staining with both insulin and pro-insulin. So if there may be actually cells or vesicles that don't have insulin packed in there, but they do have pro-insulin, and are not able to secrete, cleave that pro-insulin into C-peptide and insulin and secrete that. But if we're able to measure VMAT2 in both these pro-insulin and insulin states, then actually we think this might be more of a measure of the functional capacity, we would want to say, because there's the ability maybe to treat this population and find a drug that can get those pro-insulin islets kind of working again, and maybe there's something there. So that's why we think of it as a functional capacity, the capacity that may be available for secretion. And also these vesicles with pro-insulin are actually larger than the ones with insulin. So there may be these kind of like bloated vesicles that are packed with pro-insulin that are not creating insulin. And so you have this, again, kind of this capacity that's increasing, and we can measure this with PET if we're binding to that VMAT2. These larger islets maybe have more VMAT2 on there. So if we're taking this measure of VMAT2, we're actually saying you kind of have this increased functional capacity, but it's not going anywhere compared to the beta cell mass. So you can think of it similarly for the pro-insulin to C-peptide ratio, except we're getting that VMAT2 to beta cell mass ratio. So kind of all the non-functional versus just the functional kind of vesicles that are there kind of over the beta cell mass, this ratio.
Yeah, Jason, it kind of suggests that maybe some kind of synaptic mechanism is, being affected.
Yes.
And, as they sort of like grow larger and larger, the things can't get released. And it reminds me of some of the work that Zach Freiberg is doing at Pitt, where he's really looking closely at the neurotransmitter control and changes in, pancreatic beta cells. So that's
Yes.
he specifically is looking, at sort of like the dopamine side of things, but he does have a lot of really interesting work to kind of probe further into this.
Yeah, I've seen a few great papers already from that group. So yeah, they're doing a lot of good validation work that we're just starting to dig into. So it'd be great to connect at some point and try to work
Yeah,
together a little bit more on that. Yeah.
for sure.
Um, and then so this just kind of summarizes what I was saying in the last slide a little bit, but the idea then is that you have this dopamine synthesis, but then you have these beta cells with proinsulin and insulin kind of being packaged in there, and maybe the insulin in some cases is being manufactured. In some cases, they're getting swelled and bloated with proinsulin, but not secreting. And so you can get a kind of a measure of these vesicles within the beta cells by looking at VMAT2, but then you can get kind of a measure of dopamine receptors and beta cell mass in that case. And I do want to briefly mention this. There's a little bit of literature where there may be some binding to alpha cells of the dopamine. And there's my response to that a little bit would be that there's also evidence of these kind of transitions from beta to alpha cell in diabetes, and there needs to be a little bit more work done to try to figure out whether those are beta cells that became alpha cells or alpha cells turning into beta cells during this early stage disease, because there is some proteomics data that kind of shows overlaps of dopamine going into alpha beta like kind of crossover stages. So we don't know, we could be measuring sort of a population of those cells that may be of interest. We don't know the details yet on that, but they certainly could be part of this, say, non-functional mass that we're measuring.
Quick
And just
question,
yep, go ahead.
Jason from the chat, what is known about the gene expression of neuroradioligand targets in stem cell derived beta cells or islets, and how is this different than primary human islets?
It's a great question. It's something I've thought about a lot. I think where we really, in some cases, should be focusing as well on looking at some of those stem cell like beta cells that are sort of developed to become beta cells, and maybe find targets early on that we know are going to be in those cell populations. And then we can, if they're transplanted into the pancreas or whichever site that they're being transplanted, then we can track that using PET somehow. So I think there's still a lot to learn about some of those stem cell like populations, and really find maybe an appropriate target for those cells specifically. And then maybe you can have sort of an endogenous and non-endogenous measure of different beta cells or stem cell like beta cells, if that makes sense.
Just to follow up on that, we heard a
Yep.
talk the other day from Dan Hu at Penn, talking about his organ on a chip and all that they're doing there to kind of further investigate the sort of the stem cell milieu around, beta cell lifespan and everything else. But I think that I wondered how might your approach, your imaging approach, intersect with organ on a chip or not?
I mean, I think it would be a very good system to kind of validate, to as one of the steps to validate these tracers as well. I mean, we've done a lot of validation has been done, but especially in those early kind of stages of stem cell development. And I mean, one of the particular examples is that we can't do these yet in pediatric populations because of the radiation.
Right.
But so if you wanted to image very early on in the kind of development of the pancreas and kind of understand that growth phase of the pancreas before diabetes, in the sort of early phases, that would be where lab on a chip would be very interesting. I'm not sure the setup of what kind of tracers you can kind of flow through the system and stuff like that to see if that would, but if there's anyone on here that's doing that, be happy to connect and see if we can work on that. It'd be really, really interesting. Yeah.
Well, we have Dan coming up giving a talk soon, but I'll try to connect you with him as well. But Yeah, it's really interesting, especially when you were talking about the sort of the flux between alpha and beta cells and how can you capture that? How can you capture those changes? That just sort of came to mind, what he's putting
Yeah,
forward.
that's something that even in most reviews we come up against that question of really how do you know that it's only say X population of beta cells and not some other kind of transitionary, and we don't know that yet, but we do know that maybe those transitionary states are still valuable information from our imaging point of view. We just haven't pinned down every single subtype yet, which I mean, if people want to work together with that, I'd be very interested in trying to nail that question down. So
It's almost like you have to set up your own atlas.
yeah, exactly. No, I mean if there's a way, that'd be interesting. Yeah.
Mhm. All right, sorry.
No, that's good. That's good. I think yeah, just one more kind of slide anyway. So that the idea then is where we're at now is a lot of this data is cross-sectional. And so the critique we often get again goes back to what specific cell populations we're measuring. But a lot of that, if you think about longitudinal studies, may not matter as much because you're starting in the same person and measuring over the course of their development of diabetes. So really this, where you see the purple oval is where we want to look at the pre-diabetes and really see where this beta cell mass drops off within subjects. So some of that background signal that we're not sure about really may stay level throughout this, and we get still that picture of changes in VMAT2 or changes in dopamine, really reflective of disease progression. So really what we want to do now is try to serially image people 1, 2, 3 times within the first couple of years and really get a measurement of from this pre-symptomatic stage to the stage 3 of symptomatic type 1 diabetes. And I show this whole body picture here as well because you can see the striatum in the brain for dopamine here. So it would be really interesting to look at over the course of disease, is there any CNS changes in dopamine as well in that early type 1 diabetes trajectory? So we're starting to look at some of that as well with our whole body imaging and seeing if there's any correlations to the periphery as well. But really, we want to try to image a lot of this cross-sectional stuff has shown utility, but we really won't know if this is useful until we're able to do serial longitudinal studies, similar to some of the folks in the MR field now of type 1. They just had a great paper that showed they're able to track the first 5 years or so of type 1 diabetes and really show changes in the exocrine pancreas. And we'd like to pair that with a similar thing in the endocrine. And I think all of those together would be a powerful tool to track type 1 diabetes, kind of morphological and functional changes going on in the pancreas. That's
Fascinating.
just an
Yeah,
overview. And yeah, if anybody else has any questions or
that's great. Yeah, here's another question. Are there pediatric studies on record for other indications that use the VMAT labeling?
I'm not sure about the VMAT labeling. I know with GLP-1, there's a lot of studies with insulinomas. And that's actually some of the first studies that were showed utility in the pancreas, was that they were able to measure in pediatric populations like kind of GLP-1 expression on some of these insulinomas. I will say that some of the technology now for the PET scanners themselves is getting to the point where we can do whole body imaging and inject radioactive doses of 1/100th of what we do now. And because the machines are so sensitive and some of our image reconstruction algorithms are getting very good, we theoretically are at the point where we can get down to scans that would be the same as a cross-country flight for the amount of radiation. Those are still in the works, and we'd have to validate those a little bit more. But I could see within 5 years or so being able to do those kind of scans in pediatrics. But I mean, I recently saw that almost 40% of type 1 cases are over 30 years old now on their first diagnosis. So there is still a large population that we really don't understand and can image those folks as well. Not that the pediatric cases aren't the most significant, I would say, because it's the most rapid progression, but there's certainly a number of phenotypes that we still have yet to fully understand.
Yeah, I would ask, are there any studies you can point to that were neuroactive drugs that are used to target, sort of dopamine um, driven syndromes, that cohort of people have those people been sort of examined in this way to look at changes in, BCM and beta cell phenotype?
Not that I know of, actually, but I was just talking to one of my colleagues who is a Parkinson's researcher yesterday, and there's a lot of drug treatment studies. This is primarily type 2 diabetes that kind of has a comorbidity with Parkinson's, but they'd be interesting to look as well just to see if they're being treated with some kind of dopamine agonist or antagonist to image. They already image the brain, so we could image the pancreas as well and see if there's any kind of changes with those drugs. And then perhaps that's some kind of supplementary drug that we could use in type 1 diabetes or something. If it shows utility in kind of stimulating insulin secretion in the pancreas somehow, it might be useful. So that's the first one off the top of my head, but there certainly are other pathologies that are being treated with dopamine.
You can think about sort of the psychiatric cohort that might be
Yeah, exactly.
many people even available to undergo scans that might even be, you know, candidates for this.
Yeah, I think that the key would be to get them just before therapy. So
Mm.
sort of between diagnosis and before therapy. So then you can see if there's kind of that immediate response, not just in the brain, but in the pancreas to some of those drugs as well.
More
It'll
discussion
be,
there with
yeah,
Zach
it'll be
Freeberg.
very interesting to do. Yeah.
Okay, any further questions? This is a fascinating study and approach. I really appreciate you sharing your work with us, Jason. We just make sure anyone has their hands up
No, thanks for having me again. No, so this is fun. Yeah.
for sure. Okay, well, great. Really enjoyed this talk, and I will connect you to those we discussed offline.
Sounds great. Thank you very much.
Thanks so much. Have a great rest of your day in New Haven.
You too. Thanks.
Bye-bye.